My name is Floyd. I'm in Maria Hadjifrangiskou's lab, and we study uropathogenic E. coli, which is the primary causative agent of urinary tract infections. Specifically, I'm looking at the regulation of an adhesive fiber that is key for establishment of acute infection within the bladder, and also for the binding to and the ability to form biofilms on urinary catheters. Specifically, these results derive from some MALDI-TOF imaging mass spectrometry analyses, and so from those original observations, I've been further digging into the regulatory aspects and have found two novel regulatory mechanisms in which oxygen levels actually dictate the expression of these adhesive fibers. So it's the first time we've really gotten an idea that maybe oxygen plays a role in controlling these fibers that are crucial for urinary tract infections.
I'm Felicia Roland, and I'm in the lab of Dr. Bradley Smith at the University of Notre Dame. Our lab, we're made up of both biochemists and organic chemists. So I work on the biochemical side, and I work with these dyes over here that the organic chemists make. So we kind of focus on this theranostic idea, so we have therapy and then diagnostics. Our diagnostic aspect uses fluorescent dyes for tumor detection, so you can detect cancer. We use in vivo imaging to basically track things throughout the body. In these crokinane dyes that we use, we do a photothermal therapy treatment of cancer. So what these dyes do is they very efficiently convert light energy into heat. So you're basically able to just heat up cancer cells so much that they die. And I just did a set of experiments here showing that you have accumulation of these nanoparticles in the tumor, and then you're also able to ablate them. So I was able to show that you can basically decrease tumor growth over time and hopefully help cure, well not cure, but completely eliminate tumors.
My name is James Roland. I am here at Vanderbilt in John McLean's lab, and what I'm doing is mostly method development right now. And so we're trying to figure out how to solve biological processes, but particularly studying the metabolites. And so what we do is we get biological samples from our collaborators. Specifically right now, we get them from the cell lab, and we're looking at lung epithelial cells. We're spiking it with zinc sulfate, right? So then we do a metabolite extraction. We run it through a system, which gives us a bunch of descriptors for each of the metabolites. So because it's LC, like liquid chromatography, we get information about its solution polarity, and so it separates things based on the polarity. We have ion mobility, which is similar to running a gel, except instead of taking like an hour to run a gel, you can get the same separation in a matter of microseconds or milliseconds. And then we have mass spectrometry, which gives us information about the size of the molecule based on its charge, and then we also fragment everything, which is pretty much which is pretty much a fingerprint of what the metabolite is. And so what we do in our lab is we try to find metabolites that are either specific to the control or to the treated. Then we have developed this metabolite validation in which we use those descriptors. As we get more and more specific, we use more and more of the descriptors, and we're trying to identify what they are. That's it. I'm done.
Hi, my name is James Wepy, and I'm a third year in the Marnett lab here at Vanderbilt, and I study COX-2 metabolism. So you probably are familiar with COX-2 metabolism because anytime that you take a drug like Advil or aspirin, you're actually inhibiting COX-2, so you're preventing it from doing its job. And what we're looking for is glycerol prostaglandins, which is one side product of COX-2 metabolism. Unfortunately, no one knows what they do. So in order to figure out exactly what those biological effects are, we found the enzyme that metabolizes those. So by inhibiting that enzyme that we've just done a lot of work figuring out exactly which enzyme this is, we can build up levels of those glycerol prostaglandins and hopefully figure out their effects. So I've done some work looking at how we can control this enzyme using small molecule inhibition or genetic knockouts, and that'll prevent any metabolism of these molecules. We can build them up and find their effects.
Hi, my name is Stow, and I am in the McLean lab at Vanderbilt University, where we do ion mobility mass spectrometry. as an analytical separation technique where we can get separation in 2 dimensions, both mass to charge ratio and then size and shape and the mobility dimension. And so where my research comes in is actually generating theoretical confirmations that we can then compare back with our experimental measurements to get more in-depth structural information. So in particular, I've looked at a pretty extensive list of metabolites here, and we're trying to see if the gray bars, which represent the theoretical range, match our experimental measurements, which are indicated by the circles and triangles. And so basically, if it lines up, it was a circle. If not, it's a triangle. So we're basically just trying to almost use it as a predictor here to get collision cross-section information for future research.
So my name is Raleigh Godsey. I'm at the University of Minnesota studying bacterial sensing and signal transduction proteins. So we are interested in these because they are involved in a number of important responses such as virulence, antibiotic resistance, and chemotaxis. And so these are things that would make very good novel therapeutic targets. The way that we are looking at these is by studying the phosphorylation event here on the conserved histidine of our residues. And so my research is focused around development of specific probes that allow us to study the phosphorylation event. This has been very difficult because the phosphohistidine species is very unstable. At a neutral pH, you can protonate the imidazole and essentially hydrolyze that phosphate off, making them very difficult to study. So I've been working on different click probes to be able to fluorescently examine these activities of these proteins, and hopefully we'll be able to quantitate this and be able to study them more as therapeutic targets.
Alright, so from a zebrafish high throughput screen looking for developmental modulators, we identified a compound called EGM1. We validated it at the phenotype to associate with the hedgehog signaling pathway, and it functions at a node downstream of the typical regulator smoothen, which is typically therapeutically targeted for basal cell carcinoma, medulloblastoma, and a variety of other human diseases. We made a variety of analogs of this compound in order to identify compounds that had better solubility, better potency in the hedgehog signaling assay, and identified a variety of slightly modified compounds that improve potency and improve solubility. We tried to make some changes to the core of the molecule, but ultimately we identified 4 compounds that we're using for further development.
I'm Diana. I'm in Eichman's lab, and we study DNA damage. So my project focuses on the Huron domain, which is a domain in HLTF, which is a protein involved in DNA damage tolerance, which is one of the DNA damage pathways that we have. So I was trying to characterize how Huron is involved in HLTF function. So I was able to purify Huron, and then I determined its specificity doing binding assays, gel shapes, and determined that it binds specifically to the 3' end of single-stranded DNA. And then I was also able to determine the structure and looking at the molecular details of its interaction. I found that there was a DNA binding pocket, which allows it to bind to the DNA. I mutated the residues that we thought were important for this interaction and tested that and saw that they were all important for binding. And then I looked further into the function of HLTF, looking at assays, basically for progression assays, and determined that Huron is in fact important for HLTF function.
Hi, my name is Scott Shaw. I'm from the University of Notre Dame. I work in the lab of Dr. Bradley Smith, and in our lab we employ a mixture of biochemists and organic chemists. The organic chemists are making light-sensitive novel molecules that the biochemists then find applications for. In the past, we've done a lot of fluorescent particles and compounds for imaging, but our group actually came up with this compound that does kind of the opposite. It's very good at absorbing light and converting that light into heat. That's called photothermal. So some interesting properties of this compound is that it is absorbing red light, which is very good at penetrating through the body, so we can imagine medical applications for that. It's very efficient, and it's also very stable. It doesn't interact with oxygen, unlike a lot of those photothermal compounds out there. So my job was to come up with some potential applications for this, and as a preliminary start, I worked with liposomes. Liposomes are artificial membrane systems. That means they're like the cell membrane, only they're man-made. I incorporated this compound into them. I hit them with a laser, and that laser is able to trigger release of a water-soluble payload from the inside of the liposome. So we can imagine this system is potentially good for drug delivery applications or combination drug delivery with photothermal therapy.
Okay, so my name is Kevin Rodriguez. I'm a second-year graduate student from Professor Brandon Ashfeld's lab in Notre Dame. And so what we're looking at is basically synthetic methodology. So our goal is if people, medicinal chemists, can actually use our chemistry as they investigate the targets. So what we're looking at here is we're looking at the family of alkaloids known as spiroxindels. So a lot of these compounds, they have C3 fused spirocenter, and what we've developed is a phosphine-mediated cycloaddition approach to access these molecules. So what we're looking at here is we're using unconventional carbonyl reactivity, sort of like carbene reactivity shown here, to access these compounds. So what we've done is our first project is we can do Impelong chemistry with a 1,2-diketo ester and alkene to get to various cyclopropane spiroxindels. And we've also looked at doing 4+1 cycloaddition reactions with an orthoquinone methide precursor to get these 5-member rings. Now our application towards that is our biological assay shown here. So our whole animal assay relies on a Drosophila cancer model.
I'm Lauren Palmer in Eric Skaar's lab in pathology, microbiology, and immunology. And I'm just finishing my first year of a postdoc. And what I'm talking about today is a project I have looking at maintenance of lipid asymmetry in Acinetobacter baumannii. Acinetobacter baumannii is an important nosocomial pathogen, and it has increasing rates of drug resistance. And so it's a gram-negative opportunistic pathogen, and as a gram-negative pathogen, it has an outer membrane and an inner membrane. And in gram-negative bacteria, the outer membrane is composed of an asymmetrical lipid bilayer. So the outer leaflet of the outer membrane is lipopolysaccharide, and while the inner leaflet and the inner membrane are composed of phospholipids. So under certain conditions, phospholipids can actually accumulate in the outer membrane so that the lipid asymmetry is not maintained, and this makes the bacteria more sensitive to membrane stresses, detergents, and antibiotics. And so what I'm looking at now is one system that's involved in maintaining this asymmetrical outer membrane and how it contributes to pneumonia pathogenesis.
Hey, I'm James Dodds. I'm a third year in the McLean lab, and I focus on mass spectrometry. So basically, I try to take complex mixtures and separate them out into individual components. And so my research focuses on looking at two of the principal producers of mass spectrometers, Agilent, who made their 6560 instrument, and Waters with the Synapse G2. And I look at both of these instruments and try to run known mixtures on them and identify which system is the best for our biological applications here in the McLean.
Hi guys, I'm Mike. I'm a grad student in the Meade lab at Northwestern, and our lab focuses on applications of metals in medicine. So the particular application I'm working on is using gadolinium or radioactive metals to develop probes to study the receptor expression levels in breast cancer and other reproductive cancers. So in these cancers, estrogen receptor and progesterone receptor, steroid hormone receptors, are pretty much the two most important diagnostic and kind of prognostic biomarkers in disease. So the probes that we're working to develop have a number of applications. First is in screening. We want to, in tissues that are hard to access for other kind of diagnostic modalities, to look for tissues that overexpress them where they shouldn't be, particularly in the peritoneal cavity. Something like endometriosis, where you have uterine tissue that could be attached to your intestines or somewhere where the receptor really doesn't exist otherwise.
Hello, my name is Drew Kellum, and I'm a member of Dr. Michael Stone's lab, and my research in general is studying DNA damage, but in particular the urea lesion. And here I am studying the urea lesion by MR spectroscopy and single-strand DNA to determine the biological the chemical reason by chemical structure for the biological consequence. And by using NMR, I was able to deduce that the urea lesion exists in both the alpha and beta configuration, which can be a possible explanation why it halts replication, because other alpha anomers are able to block replication. So that's a summary of my research.